Welcome to this fourth video in the series of biological molecules. This one's all about lipids. And lipids are an extremely diverse group of molecules. It includes fats and oils and steroid hormones. And the diversity is so enormous that it's hard to understand perhaps why they form a single group or why they're all put in a single group until you realize that they're all hydrophobic. These molecules are defined, lipids, are defined as those molecules which are hydrophobic. So let's get into it a little bit. With carbohydrates, we recognize that they're primarily just carbon, hydrogen, and oxygen, and lipids are no different. They're carbon, hydrogen, and oxygen. Those three atoms combined. But with lipids, there's a few of the lipids that have a few other kinds of things in them, so, excuse me, a few other atoms in them, so we'll get to that. But for a starting point, let's consider a type of lipid known as a fatty acid. Fatty acids, lots of different kinds of fatty acids. What they are, quite simply, is long-chain carboxylic acids. Now, if that doesn't mean anything to you, look at this. We've got a long-chain carboxylic acid. They've got a C double bond O, OH end on them. That's a carboxyl end, or the acidic end. It's acidic because this hydrogen can be released, so anything that releases hydrogen makes it an acid. So what we have is a long carbon chain with that kind of an end on it. And I'm just going to go like this, dot, 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 and I'll end it like that. If you remember now, also carbon is supposed to have 4 bonds around it. Well, that's satisfied here, 1, 2, 3, 4, not satisfied anywhere along here, until you realize that the rest of this thing's all just filled up with hydrogen. So we have this. All these bonds. Now look at each carbon has 4 bonds. There's bonds in here, I didn't draw them. And they're filled with hydrogen. There's quite a variation in these things. As you can see, I went dot, dot, dot in here. Well, how long are they? Well, they could be 16 amino acids, or 16 carbons long, or 18 carbons long. And that's one of the ways in which they're so variable. The reason they're hydrophobic is because they're nonpolar. There may be a little bit of polarity in this end because there's an affinity for the electrons between, a different amount of affinity for electrons in those atoms, but most of this molecule is nonpolar. What that means in terms of its physical structure, or physical nature, is that these things float. You know that what we did in the other video, if you take water in a glass like this, and you put some oil on it or some fat on top, it's going to form a layer on top. Well, these things float. They're hydrophobic. They do not mix. So there's variations in this thing because of the length. That's pretty obvious. But in addition to that, there's another variation because they don't have to just have single bonds in them. We can satisfy their bonding requirements for carbon by putting double bonds in here. So let me just take this one out here, or add another bond in here, and take this hydrogen out, and take that hydrogen out. Now you can see the bonding requirements are still there. 4 around that one, 4 bonds around that carbon, so that works. This is a different fatty acid. It may be 16 or 18 carbons long, just like the original one was, but this one has less hydrogen and it has the presence of a double bond. This one, like this, when it doesn't have all the hydrogen possible in it, it's called unsaturated. So it actually lacks possible some hydrogen and has double bonds. Saturated fatty acids are full of hydrogen. They do not lack any hydrogen. And they do not have double bonds. So this one is an unsaturated fatty acid. And you can see the variation that's possible here again is that this unsaturation point here, the double bond could be here or here. It couldn't be here, there'd be too many bonds on the carbon. Or it could be anywhere along the length of however long this molecule is. So like I say, there's a tremendous number of tremendous variation possible in these things. And there's something else that's possible. This arrangement right here is known as a trans arrangement. Because the two hydrogens are across from each other, and the rest of the molecules are across from each other. I can draw it a little bit differently, talking about unsaturated ones. They could be cis or trans. So unsaturated fatty acids could be cis or trans. And cis, here's the double bond, we just look at the double bond. Cis means that the hydrogens are actually on the same side, and the rest of the carbon chain then is like this. Whereas you can see with the trans configuration, if I just draw it again here, the trans configuration, the hydrogens are opposite to each other, and the rest of the carbon chain is like that. And you've probably heard of trans fatty acids, trans fats. They're not so good for you. Right? So these possibilities, these things are all isomers of each other. Isomers are molecules that have the same atoms, they're just different arrangements of them. Now there's one other thing that we can mention about unsaturated versus saturated fatty acids. Saturated fatty acids, here's a way to remember it, tend to be more solid at room temperature. Unsaturated fatty acids, like these ones, tend to be more liquid at room temperature. Saturated fatty acids are those which tend to be in animal tissue. Unsaturated fatty acids tend to be those which are in plant tissue. So you talk about vegetable oils and that sort of stuff. Those things have the unsaturated fats in them. So like I say, there's a lot of variation just in fatty acids alone, but lipids as an entire group of molecules is a lot more than just fatty acids, because fatty acids tend to bond onto alcohols. Now let me show you what that looks like. I'll erase this, I don't need it, I can redraw the part that I do want. One of the alcohols which is very common with fatty acids is an alcohol called glycerol. Now glycerol is a 3-carbon molecule. I'll draw it vertically like this on purpose, and alcohols all have OH, the hydroxide groups on them. That's the functional group of alcohol. So this glycerol molecule is a 3-carbon, 3-functional group molecule. That's glycerol. Fatty acids bond onto glycerol. The way they do it, and this is going to bring back a memory from the carbohydrate topic, here I'm going to turn the fatty acid around. There's the carboxyl group I mentioned, but when I had it drawn before, it was on the other side of the molecule. And you can see what's going to, I won't finish this, you can see what's going to happen. These things are in close proximity with each other. Look what's going to happen here. They bond together, a molecule of water is released, an ester bond is formed. Remember, the ester bond is the one that goes from carbon to oxygen to carbon. The same as we had when we had a disaccharide, because when glucose bonded to glucose, we got maltose, but the two parts of the maltose molecule are held together by an ester bond. Now, right away you can see, okay, that opens the door for a lot more variations, because I just drew it bonding to this one. It could be here. It could be here. Or you could have 2 of them. Or you could have three of them. The number of fatty acids, there's 3 bonding sites, they could all be filled. More variations, because these carbon chains on the fatty acids could be saturated or unsaturated. And if they're unsaturated, the double bond could be in different locations. So you start adding up the variables, there are literally thousands and thousands of possibilities for different types of these molecules. Now what are these molecules called? They're not fatty acids anymore. These things are called neutral fats. Glycerol plus fatty acids. You could have a monoglyceride. Now right away you can probably figure out what that is, because one of the glycerin functional groups is occupied by a fatty acid. Or you could have a diglyceride, or you could have a triglyceride. Different variations possible. So what's the importance of all this stuff? These things? It's an important secondary source of energy. We have them in our bodies. I mean, with subcutaneous fat, that's what this is, neutral fat. It's a subcutaneous fat under our skin, subcutaneous. It's for insulation, keeps us warm, protects us from damage. We can metabolize lipids fairly easily for a source of energy if we had to. Although we do tend to use glucose first. So, like I say, there's a tremendous number of variations of these. There's even another possibility, and this possibility shows up in cell membranes, which is an entire topic that we get to in a different video, but I will introduce it here. It's where one of these, and I'm just going to, for the sake of the diagram, I'll erase that so you can see what's going to happen here. I'm going to change the side of this thing here. So I'm going to put the OH on this side and the H on this side, and we can bond another O to C. Now remember, the double bond O is still there. So we've got a diglyceride here. I won't put the hydrogens in, but what happens in some of them is this, instead of bonding onto a fatty acid, in here, we wind up with a phosphate group bonding onto here, phosphoric acid, and a choline group, which contains nitrogen. So what we have now is a very different molecule, very significant in cell membranes. This part here is very polar. This part here is very nonpolar. So you can see how important it is. This forms an interface between polar substances and nonpolar substances. Very important in cell membranes. This thing is called a phospholipid. And it's simplified, if we're drawing it, it's simplified as a polar head. So this is the head, which is polar, and two non-polar sequences that come down from there. So if you see these little things that look like, I don't know, little organisms with two tails, that's a phospholipid. So that's just another variation of lipids. We've got all these fatty acids, we've got neutral fats, we've got phospholipids, we've even got more complicated ones. Before we get to the most complicated one, I should mention that wax is one of these kinds of molecules. You know yourself, you take wax and put it in water, it floats, it doesn't mix. Even if you grind it up small, it will not mix, because wax is a combination of fatty acids and alcohols like glycerol, only larger than glycerol. So these things are fatty acids plus long alcohols. Now you think about all the different kinds of waxes there are, whether we're talking about sealing wax for canning, or whether you're talking about earwax, or you're talking about beeswax. There's lots of different kinds of waxes. So again, the number of variations in these molecules is tremendous. The thing that holds them all together is, generally speaking, they're all non-polar, at least this part is, on this one, that one's polar, and they don't mix with water. They're hydrophobic molecules. The last one, the most complicated one, I can get rid of all of this now, are steroid hormones. Things like estrogen and progesterone and testosterone are steroid hormones. And these things are non-polar, they're lipids, and they all share a common structure. So if you see this structure, these are ring structures right now, and I'll just clarify what that means in a second. Five-cornered ring here, 6, 6, and 6. Every corner here has a carbon on it. So you can see the bonding requirements are mostly filled for these things. You've got to put a couple of bonds in here and one more bond in there and here like that, right? So it's a cyclic combination of carbon and hydrogen. Obviously it's not going to mix well with water. There's nothing here to be polar. But the differences between testosterone and estrogen and so on is there's little side branches. So instead of having just hydrogen out here, there may be something else bonded to it, or there may be something else bonded to it down here. So these things are called steroids. And we're talking about things like estrogen and testosterone. Steroid hormones. But there's even another one that is the same basic structure. It's not a steroid hormone, it's cholesterol. And we've all heard of cholesterol and saying, oh, high cholesterol is not good for you and all that kind of stuff. That's because it doesn't mix with water. It's a shape of a molecule that looks like this. It's extremely important in cell membranes, which is a topic we have to get to. Our body makes cholesterol, delivers it through our circulatory system, but you don't want too much, because too much of it is not a good thing. So that's the most complex type of lipid that we have to look at. Just before we finish, I want to go back to this diagram from previously, where we take the unit molecules, the monomers, and we combine them to make a polymer. I've got 5 things there, so I've got 5 things here, and that sort of thing. We did this with the fatty acid bonding onto the glycerol molecule, releasing the water. The thing is here, lipids are not true polymers. These are the monomers. These are polymers. The diagram works. It helps understand what's going on here, but lipids are not true polymers simply because there's an extremely limited number of pieces you can put together. Poly means many. And if we've got glycerol, you've got glycerol plus possibly 3 fatty acids. That's it. There's only 4 molecules. You can't get hundreds bonded together like you could get hundreds of sugars bonding together to make starch and glycogen, and hundreds of amino acids bonding together to get proteins. So these are not true polymers. Proteins, that's our next video. Thank you very much. And as always, if you'd like to subscribe, please click on the gecko.